Welcome to an edition of CCSD Living Environment. In the last video we talked about fertilization and development, and today what we're going to look at is patterns of embryological development. How do the embryos actually develop once they've been formed? So after fertilization, the zygote goes through a series of mitotic cell divisions. Remember, mitosis, identical copies, genetically identical. So it goes through a series of mitotic cell divisions called cleavage, which is cell division without growth. So every cell division results in cells that are half the size of the original cell. So each time the cell divides, the overall structure isn't changing, but the cells that are dividing inside are getting smaller and smaller and smaller. Remember, the zygote is a fertilized egg cell, and so what we have here in our first picture, and it tells you what structures you need to label. You can see my cell membrane, I have my nucleus and my nucleolus, and then going around the outside is a structure we haven't talked about before. That structure is called the fertilization membrane. The purpose of the fertilization membrane is to prevent other sperm from entering the egg after the first one has gotten in. So after the first sperm cell enters into the egg, we don't want any more sperm to enter. That's enough DNA. You have half from the mom in the egg, and now half has been contributed with that first sperm. No more sperm need to enter the egg. So a membrane goes up to prevent further sperm from entering that egg. So that takes us to what they call the two-cell stage. It's the result from the first cleavage, or that first mitotic cell division. Notice what I have here is two cells, and they're smaller than what that last cell was. Again, the cell membrane is going around the outside. We have the nucleus, the nucleolus, and what can we label now that we've talked about during mitosis? And that would be the cleavage furrow. And you can see that's where that cell is starting to pinch in. The total volume of this cell is not changing. Remember, cleavage is division without growth. So the total volume of that cell is not changing. It's not getting any bigger, it's not getting any smaller. That takes us to the four-cell stage. Four-cell is the result from the second cleavage, or the second mitotic division. And again, notice that the cells are smaller, and they're about half the size of the cells that were here. So for each one I have a cell membrane, for each one I have a nucleus, I have a nucleolus, and again we could label the cleavage furrow in each location where the cells are starting to pinch away from each other. So the question then is, does the volume of each cell change? And the answer to that question is yes. The volume of each cell does change during cleavage. As the cells divide, they have to get smaller and smaller and smaller to fit into the whole volume. Remember, the total volume isn't changing. So if I'm going to keep dividing cells, then each cell has to get smaller and smaller so they can fit into the size that I have. Because that size doesn't change, so as each cell divides, they have to decrease in volume. Next we come to the 8-cell, which would be the result after the third cleavage, or the third mitotic division. There's a picture of the 8-cell there in your note packet. It's very difficult, as you can imagine, to draw three-dimensionally 8 cells in a circle. So if you would please label the cell membrane. You can insert a nucleus and a nucleolus into one of the cells, and you can also label the cleavage furrow in those cells as well. So what happens? Well, by the time we hit, say, 30 to 40 divisions, there becomes too many cells to keep counting. So at that point they change, and instead of naming the cells, they call it the morula. The morula is a solid ball of cells. So in that three-dimensional sphere, it's full of cells. Each cell has a cell membrane, a nucleus, a nucleolus, and the cleavage furrow, again, is where each one is pinching to give cell after cell after cell. So at that point, they no longer name it by number of cells, they just call it the morula. So the morula is just a big ball of cells. Well, what happens next is kind of interesting, because all of the cells somehow find their way to the outside, and we end up this rearrangement with a hollow ball. Think about a ping pong ball. So the layer of cells is the plastic around the outside, and it's hollow in the middle. This is hollow in the middle. So what I end up with then is the cell membrane, the nucleus, the nucleolus, and the cleavage furrow. But the interesting thing with the blastula is that it's only one cell thick. There's only one layer of cells going around the outside, as opposed to here where the whole thing, it was a solid ball. So from the morula to the blastula, the cells rearrange, and now I have a hollow ball of cells. So that takes me to a process called gastrulation. Think about that ping pong ball that we had. What I want you to do is think about holding that ping pong ball and taking your thumb and indenting the ball. So you're taking that ball and you're indenting it. Remember, it was that hollow ball, the blastula was the hollow ball, and we're going to indent that ball. So that process is called gastrulation. In gastrulation, it's the process that produces a structure called the gastrula. So again, here was that hollow ball, and what did I do? I indented the ball. So what distinguishes the gastrula stage from the blastula stage? That indent. The indent is the difference between the blastula and the gastrula. And the process to form the gastrula is called gastrulation. So what stage does differentiation actually begin? Well, what is differentiation? We'll get to that in just a second. But where does it start? The gastrula is where the cells start to take on their function and form the three primary germ layers. Some totally new vocabulary here. So what does different mean? Well, different means different, right? Okay, well, what does different mean? Different means that they're not the same. So how, in other words, do we go from all of these cells after mitosis? Remember, identical cells, identical cells, identical cells. That's mitosis. How do we go from mitosis to getting our liver cells, our kidney cells, our lung cells, our heart cells, our brain cells, our skin cells, our muscle cells, our hairs, all the different cells that we have, they're all different. How do we go from all the same in mitosis, cleavage, to different cells that make the organism that we are? That's the process of differentiation. And those cells starting to become different things starts to take place during the gastrula stage. And that's because we start to develop these different layers called germ layers. The first germ layer is called the endoderm, endo being the inner layer. The endoderm gives rise to the lining of your digestive and respiratory systems. Think about the most inner part of you. What would that be? Well, that would be the inside of my digestive system. Not just the inside of my outer cavity, but the inside of the inside. And that would be the lining of the digestive system and the lining of our respiratory systems. So that's what the endo or inner lining is going to form. And then I have the ectoderm. Ecto means outer layer. The outer layer gives rise to our nervous system, which would be like your brain and spinal cord, and your skin. Well, it would make sense that the outer layer gives rise to my outer layer, which is my skin. Okay, so that one's not too bad. And then that takes me to the mesoderm. Mesoderm is the middle layer. We've got this M thing here going on. So in that middle layer, it gives rise to the muscles, your reproductive, excretory, and circulatory systems. Remember, your excretory system is your urinary system, basically. So what we're doing with the mesoderm, we're putting the stuff in the middle. Well, what's in the middle? Well, my muscles are in the middle. What else? Well, my reproductive organs are in the middle of everything else, as is my circulatory system. It's kind of in the middle of everything. So that middle layer, that mesoderm, forms the cells for the stuff that ends up in the middle. So the definition for differentiation. Each new unspecialized cell, remember after mitosis, it was identical copy, identical copy, identical copy. So each one of those unspecialized cells is modified and specialized as the cells multiply. Different sections of DNA are turned on and off, resulting in different kinds of specialized tissues, cells, and organs. So think about that for a minute. If it's cleavage, if it's mitosis that produces each one of those cells, that means each one of those cells has exactly the same DNA. So that means my heart cell has the same DNA as my kidney cell. Well, my heart and kidney don't perform the same function. So how can they have the same DNA? Well, that's because some of the DNA in the heart cells get turned on to be heart cells, and that same DNA in the kidney cell doesn't get turned on. Instead, the DNA in the kidney cell, to make it a kidney cell, gets turned on. And that DNA does not get turned on in the heart cell. So in the process of going through and basically deciding which cells are going to become what things, there's this process of turning on or turning off the information. And that's the process of differentiation, which, remember, takes place or starts to take place during the gastrula stage. At this stage, the embryo is very vulnerable to alcohol and drugs because organs are just starting to develop. This is the point along the way where things haven't really been told what to do yet. The cells are so new, they're so fresh, they're just getting started to become whatever they're going to become. This is when those cells, this is when that developing offspring is so sensitive to things that could potentially affect them, like drugs, alcohol, tobacco. Any of those things that mom is taking into her body at that point have a severe effect, or can have a severe effect, on the offspring or the developing embryo at that point. All right, so let's take a look then if we put it all together. If I start out here with my zygote, that's letter A. The zygote, remember, is the fertilized egg cell. That's what happens when the sperm and the egg come together. I get the zygote. That is a 2N cell. It has all of the information to become the organism. At that point, cleavage begins. So cleavage starts right after fertilization. That gives me my 2 cell, my 4 cell, my 8 cell, and that, remember, is going to give me that solid ball of cells. From the solid ball of cells, they rearrange to give me the blastula, and then gastrulation takes place, the indent, to give me the gastrula. This is where differentiation is going to take place. So each cell becomes what it's going to become, and I develop my new organism. Whether it's a human or a starfish or a clam, an oyster, a whale, an octopus, a squid, a giraffe, an elephant, a lion, a zebra, I don't care what the organism is, a maple tree, algae, pick an organism. The process is very similar in the fact that when differentiation takes place, that's when you're going to end up with your new organism. The genetic information in the zygote, so the genetic information that I have here in my zygote is exactly the same, completely genetically exactly the same as the information that I'm going to have in the embryo and that I'm going to have in the adult. All of that information is exactly the same. So again, what is an embryo? An embryo is an organism in the early stages of development. So in that very beginning, we've gone from zygote and we've gone through cleavage. Once we get past that point, okay, once we get to that point, that's where we have our embryo. And that embryo is going to start to grow. That's where growth begins. The organism is going to start to grow to give you whatever organism it's going to become. So that genetic information, the DNA, all of the chromosome information is the same from the zygote to the adult, to me. So the genetic information that was in the zygote when the sperm and egg came together at fertilization, that genetic information is exactly the same as the genetic information that's in all of my cells today. And that's the whole idea that you need to understand, is that when we undergo cleavage and we just produce all of those cells, that's how we go from that single fertilized egg cell to a multicellular organism that we are today. That takes us through the patterns of embryological development. I hope you picked up something from today's video, and I'll catch you next time.